Hi, and welcome to Cell Biology Review for Biology 476, Topics in Stem Cells. So we're just going to talk about some cell bio that is relevant to stem cells and to this class, so that makes sure we're all on the same page. So as we know, the cell, a single cell, unicellular, organisms can be multicellular. You can be a prokaryote or eukaryote. We are eukaryotes, so this is us. So we're going to mostly be talking about mammalian or human cells. As we know, the prokaryotes, they don't have a nucleus, they don't have the organelles inside. They're like a bacteria or an archaea. So we're just considered useful eukaryotic info. So we're not going to memorize stuff about the prokaryotes. We know they're there. And we're going to think about eukaryotes, right? The membrane bound, has a nucleus, the control center, all of that. We're not going to talk about organelles up front, but we will talk about mitochondria as we go through the course, and we'll review stuff anytime something comes up. So let's go. So our organelles, as we know, we can always come back to this when we need to look at stuff. Again, we're going to be talking about mitochondria, right? Guy that does energy, ATP, cellular respiration, that kind of stuff. We know the nucleus, right? This is the guy that has our DNA. Super important. And again, like I said, the rest of the stuff, when it comes up, you can always refer back to any kind of text for organelle stuff. Cell multiplication. Well, cell division, right? We had to start as a single cell. Some organisms stay as a single cell. Anything that's eukaryotic, in order to replicate, whether you're unicellular or multicellular, the cell goes through mitosis. And each cell has some sort of finite lifespan, right? We definitely replace dead and damaged cells by mitosis to make sure we don't lose too many cells as an adult. So let's take a look. If we remember the cell cycle, it is composed of interphase. So in our little diagram, that's the orange, I guess that would be, and mitosis. And here we show that in yellow, right, the M. And so what they're telling us is that, right, here's M, it's just a little part of the cell cycle. The rest of it is considered interphase, like I said, in orange. And then interphase is broken up into several phases, or stages, I should say. Don't use the word phase, right? Mitosis is the one that has phases. Interphase has stages. That's just so we don't get confused. Doesn't really matter what you call them. So G1, growth, getting ready for DNA synthesis. Make sure you have all the enzymes necessary. S, synthesis, right? DNA rep. That's where replication happens. And then G2, growth, check everything out. And then mitosis, where we actually split the sister chromatids apart, right? So we start with a chromosome here in G1. Here we're working on making another one. G2, we have duplicated chromosome connected together. And then in mitosis, we end up with, right, 2 cells, each with one of those sisters in it. And so this little picture here is just showing us that cells can terminally differentiate, so they can get released from this cell cycle. So that'd be like you are a neuron, you're done. You're not going to make any more, right? That's it. And the other one doesn't have to do the same thing. That guy could keep going and continue through the cell cycle. One of them could differentiate, one can stay a stem cell. Ooh. Or both can stay stem cells if you need more of them. Or both can terminally differentiate if that guy's done and it's over. This little picture over here is just the cutie little guy for the phases of mitosis, right? Can we see any of these guys? This has got to be prophase 1, prometaphase. This one's got to be metaphase. This one's going to be anaphase. This one's going to be telophase. And this is cytokinesis. Right? They've broken up all the prophases into multiple steps. Prometaphase, prophase. I don't even know. I don't care. We're not going to worry about the phases. We need to understand the cell cycle to understand how stem cells work, but that's about it. So again, replication of cells. If we want to actually reproduce the entire organism and we are multicellular, we like meiosis to create gametes, right? And so then we do fertilization, blah, blah, blah. So as people, to reproduce, right? So here's our cell. We replicate, get sisters on each one. In meiosis 1, homologous chromosomes come apart. So one of each pair, and then in meiosis 2, the sisters come apart. Whereas in mitosis, right, we just start with these guys and we replicate. They show the, blah, blah phases. And then, boink, sisters come apart. So the original parent, 2 identical daughters. In meiosis, original parent, 4 not identical, right? This is N. This has half the chromosomes. This is 2N. Diploid number. 2N, 2N. Fascinating. Oh, and then fertilization, right? One gamete, that's N. Another gamete, that's N. Oh, 2N. Now you're a zygote. Oh, so what is the lifespan of a cell? It depends on the type of cell. If you're rapidly reproducing, what's our skin, hair, intestine, right? Because they're in contact with the environment, lots of them die and they need to be replaced. How about these guys? Not so much. We don't really go through too many of those guys. Most cells can have the ability to divide up to a point, but at some point you get old and you just stop, and that's called aging. And if all your cells stop and then die and don't replace, then you're dead. It's a little, not such a pleasant thought. If you divide indefinitely and you go out of control, that's what cancer is. Cell differentiation. So what happens after the zygote? Well, we have to make all the different cell types in the body. So we have to go through a lot of mitosis to do that. And then some of the cells in the cell cycle will go off and terminally differentiate. Others will keep reproducing themselves. So a less specialized cell becomes a more specialized cell, right? So anything that's called a blast is not differentiated. So a neuroblast would be like a precursor to a neuron. Lymphoblast would be an immature lymphocyte. So, right, from stem cell on. I didn't know. I felt like there should be more than that. 212 differentiated cell types. And it's thought that differentiated cells cannot backtrack to regain properties of the cell it came from. This is true in nature, as far as we know. In our physiology. It is false in lab, or what we can do something that's not natural, unnatural, something we manipulate. And we'll get more to that later. That's what this whole class is about, people. Okay, so here's our little example. If you are, here we go again, here's our little egg and our little sperm. Oh, together we make our zygote. And then that has to become everything. So lots and lots of cell divisions, and we have to make blood, bone, glands, nerves, skin, right? Everything. All the epithelial, the retina, the liver, everything came from that one zygote. So lots of differentiation. And then if you think about just any old single cell, right? Somebody in here, if they do mitosis, right, one daughter might get signals have a specific gene that's turned on that turns him into a neuron. The other guy has a different gene that gets turned on. Epithelial cell, right? Because all cells have the same DNA, right? 46 chromosomes. There's no difference in genes in any cell type. Whether you're a brain cell, that's kind of stupid, a neuron, sorry, I guess I'm not teaching non-majors. If you're a neuron or a skin cell, same genes. So how are they different? That's the $1,000,000 question. They just turn on and off a different set of genes. Not all of our genes on our 46 chromosomes, I think we have 20,000 genes. Not all of them are on or all are off. 10,000, 15,000 different combinations of different genes give us different types of cells. So it's all about gene regulation. Oh, I guess first we have to, before we talk about that, we have to talk about controlling cell division and differentiation. So it's a whole bunch, well, I guess we're talking about now, some gene regulation and also some pathways of cellular proteins. So like signals, you can think of them like hormones, signaling cells to change or become one thing or the other, turn on a gene, make a certain protein, drives a cell towards the lymphocyte lineage or white blood cell lineage versus a neuronal lineage versus a skin or epithelial lineage. Anytime these are screwed up, that's when bad stuff happens. Bad stuff is bad. We want to keep all these pathways nice and clean and under control. So your lineage is just the type of cell you become. And so you follow a path. Every cell follows a specific path from embryonic type cell, we'll call it a stem cell, to whatever is terminally differentiated. Terminal diff. That means the last stage. Stop here. Stay that forever. And so there's lots and lots of intermediates. In order to make this happen, if we want to take, scientists want to take a stem cell, and we want to make it into a neuron, we have to know all of these steps. It's not one, it might be 50. Whew. So in order to do anything with regenerative medicine, to use stem cells to make tissues or cells for any kind of therapy, we have to know everything about all of these steps. That all comes from basic research, right? Where you're just studying molecules. Scientists may spend their whole life studying this step right there, or one enzyme. But it's super important because the collective knowledge can put together this stem cell to neuron, and holy cow, you have a spinal cord injury, but they give you some neuronal stem cells and your spinal cord regenerates. That has worked in mice. And they walk again. It can happen. Oh, well, speak of the devil. So let's just zoom in, shall we? Oops, can't see very well over there. So this, we're starting with neuroepithelium. So this, we're still way back from embryonic. Right, so there's lots of other steps here. But we're going to start with at least the germ layers. And then we call that a multipotent stem cell because it can be multiple things. Can't be everything because it's already neuroepithelium. This guy, if we start back here by the zygote, this guy can be everything. So we call him pluripotent. Can't spell. He can be everything. With one exception. Okay, and so then we call the next step would be progenitor. They call this bipotential. Why? Because it can become two different things. So if you went here, if this cell got the signals to go here, turned on genes, turned off genes, right? No more mitosis, and then it just differentiates gene expression, cell signaling becomes a neuron. Woohoo! Right? If this guy went different genes turning on and off, glial, this guy then could go 3 different ways, and then this guy could split again into 2 different ways, and you can see differentiation, genes on, genes off, genes on, genes off. This guy, oh, he could get to here, but oh, he could also turn the corner. Turn on a gene, turn off a gene, get a cell signal. Ah! Hematopoietic cells, blood cells, right? Where is this happening? In our bone marrow. Is this happening now? Yes, now. All your life. Or you're dead. Right? Hemocytoblast. I can't even say that. This guy, again, not zygote. Zygote, way here, a gazillion steps to get to this guy. He is multipotent again, because he can become lots of things, but not everything. This guy here cannot go back and cannot become a neuron, cannot become any other type. In nature. Naturally. In the lab, as it turns out, we can make it become lots of things. But we're not talking about that right now. They were talking about the real world, in your body. Da-da-da-da-da. You need these. What do you think those are? Those are red blood cells. And then here's a bunch of white blood cells. Right? All these guys. Fight disease. Need those guys. And what are these guys? Platelets. Don't want to bleed to death, do ya? So again, lots and lots of signals. Genes on, genes off, genes on, genes off, genes on, genes off. All different to get those things. Again, all the same chromosomes, all the same genes, just which ones are turned on and off. Ooh, our central dogma. So of course we have to remember that. Let's zoom in. DNA makes RNA, makes protein. The only way we can get to protein is through RNA. And in Euks, we store all the information in DNA. This is our messenger, mRNA. And then we get our proteins, right? And this right here, that's just replication. The central dogma, DNA to RNA to protein. Remember we talked about differentiation again. Well, gene expression, it's what's turned on and off. That tells you whether it's going to, the same exact cell, these two identical copies of DNA, identical, same as this guy, same chromosomes, same genes. Turn on a different set over here, get a neuron. Something else, epithelial. Over here, liver. Over here, skin. Over here, kidney. Over here, I don't know. Toenail. No, that's not a cell. Whatever the cells are that spit out the keratin that make your toenails. Oh, we got to regulate that gene expression, right? It can't just be willy-nilly genes turning on and off. Otherwise, it would be just a blob of junk. So lots of places we can control gene expression, whether it's on or off. We might make an RNA, but we don't make the protein. So as long as you don't have the protein does the function. That, right, solves your problem. So there's just different ways of controlling what's expressed and what's functional. So transcription is the big one, right? That gives us the RNA, this dogma to protein. But we can also block the protein from happening. We can knock out that RNA after we make it. But that's a big waste of energy. So most of the time, if you transcribe something, we're going to translate it and we're going to use it functionally. But these other levels, we can still control gene regulation. But this guy's the big one. And again, it's just which cells are on, which genes are on and which genes are off. This is showing you all the different controls, right, in a little flowchart. Yay! But again, that's the big guy. We talked about, okay, we can go down a different pathway for what kind of cell we're going to become based on what genes are on and off, but also about cell signaling. So again, that's factors, hormones that tell cells to do something, chemicals, right? And so what happens We bind some receptor, it goes through a million different signal transduction pathways. At some point, it affects gene regulation to turn something on, turn something off. So, if we want to control division and differentiation, crazy stuff like this, which we're never ever going to memorize, all of these different craziness things, but super complicated, right? Always a bunch of steps from the binding all the way down to the actual turn genes on, turn genes off. An example of that would be, here is our embryonic cell. Let's zoom in. Whoa. No, not that. Whoa. Okay, so this would be an example from the very beginning. So this might be one of, here's zygote over here. And then, zygote does some mitosis and we get a ball of cells. Right, before anything happens. We call that the inner cell mass. Whoa. Part of the blastula, which you're going to learn about in our video. So we do mitosis, cell division, and then in this example we have a regulatory protein transcribed. Translated makes a protein. So that expression of that protein causes this cell to be different than this cell. Same DNA, identical genes. It's just this one turned on a gene and this guy over here didn't. So now they're already different from each other. So when this guy splits, these two are still identical gene-wise to this guy and to that guy and to that guy. But now this one, gene 2 turns on, but this one, gene 3 turns on. Over here, gene 1 turned on. And then when this guy splits, this guy has 1 on from its mom up here, and then turns on 2. So 1 and 2 are on in this guy, 1 and 3 are on in this guy, only 3, only 2 over here. And then when this guy does mitosis, 2 and 4, 2 and 5, 3 and 4, 3 and 5, 1, 2, 4, 1, 2, 5, 1, 3, 4, 1, 3, 5. These are all now different types of cells because they're expressing different genes. So maybe this one is that neuroblast that becomes the neurons down that pathway. This one might be the lymphoblast. This one might be a kidney blast. I don't know what nephroblast. Right? So all of these are going to become some other cell type. It's amazing. From the same thing. Oh, it's awesome. All right, that's our little review. Cell biology. Yay. Thanks for listening. Love you guys.